All right, so I hope you guys are ready to start talking about the prokaryotes. Now, prokaryotes include two domains, the domain bacteria and the domain archaea. So what we're going to do is look at the big picture of prokaryotes. I want to give you guys an overview of them, kind of what they are, some of the key things to remember. Those of you who will be moving towards a microbiology course, I want to make sure you guys have a little bit of a foundation for that because you're going to spend a lot of time discussing prokaryotes in micro. So we're going to spend some time here just, again, looking at what are the general features and characteristics of prokaryotes. But a couple things to mention. Let me get a text box going here. Some kind of key things that we would really will be beneficial for you guys to remember. Some key things about prokaryotes. They are the oldest form of life on Earth. All right, they've been around longer than any other life form that we know of. With all of the knowledge we have today, scientific evidence says these guys were here first. They're pretty much the simplest and the most abundant form of life. Now, because they're simple, don't think that they're not good or successful. This stuff's been around longer than anything else. Obviously, it's successful. Keep it simple is a mantra a lot of people will use. They kept it simple and it's functional. Human life may cease to exist. Odds are prokaryotes will still be around. Generally, we think of them at the bottom of the food chain or the bottom of the trophic pyramid. Well, when the top levels of the food chain collapse, bacteria don't care. They're still doing okay. And consider how many species humans have pushed into extinction. We still can't get rid of some bacteria that cause problems, that cause health issues, that cause disease. We just can't get rid of them. So they're pretty darn adaptable. So there are some great things about the prokaryotes as well. So we're going to highlight those things as well. I want to talk about, here are the problematic ones, those of you heading towards healthcare, medical fields, dental, etc. But also, here's the really good things about bacteria and archaea and the prokaryotes. Those of you going into conservation, environmental science, you want to find the good ones out there and use them to help improve ecosystems and things like that. Okay, so again, general things, we could also say they're pretty much, they live pretty much everywhere. Oh, I gotta get my spelling down. And live. Okay. So prokaryotes can live everywhere that we can, and even some places we can't. So we'll talk about that as we get into the diversity of the prokaryotes. Imagine living inside of a volcano. Wouldn't work real well for us, but these guys, they can survive there. So, okay, so I want to change this term here. They are primarily single-celled. All right, so you're going to sometimes see unicellular out there. Not the best term. Single-celled is a little easier to work with and usually more appropriate. So when we're looking at prokaryotes, so we're going to talk about the bacteria first, then we'll get into the archaea. They're single-celled. Now they will often form colonies where they group together, but they still retain individual functionality. They are not dependent upon another cell for their function. So that is a huge characteristic. Let me put the little star here that I want you guys to remember. They don't need another cell to function. That's supposed to be a star. Sorry. They don't need another cell to function. They can function on their own. That is why a bacterial infection is such a pain to get rid of. You knock out 99.9% of them, you left behind a few, They function, they reproduce, they rebuild their population, and sometimes they come back mean and mad and just immune to the antibiotic we used. Again, we'll get into that in a little bit here. Size-wise, you got a spectrum, 0.5 to 750 micrometers. So some of them, or many of them, are smaller than eukaryotic cells, meaning they can actually fit inside a eukaryotic cell, but there are some that are actually bigger. They're large compared to eukaryotic cells. So we have this big spectrum of diversity in size when we look at bacteria. When we go across the features of bacteria, we often see all the diversity of bacteria have the same core features, but then what makes one different than another are how they kind of tweak and adjust some of those core features. So the first exposure I want to give you, just kind of the whoa of how abundant bacteria is, take your fingernail, scratch your tooth. Hopefully you guys did that. Okay, look at that little filmy stuff there. That's called a biofilm. That's what you're looking at. That biofilm, this purple stuff, or blue stuff, a little green color to it, that's biofilm. That can come off of your tooth. That could be sitting on your pencil. That could be sitting anywhere. Those are bacteria. The stuff on your teeth is why we want to brush our teeth after eating, why we floss and go to the dentist, because that bacteria can lead to tooth decay. But they're that abundant. They're everywhere. It's estimated we have more bacteria in our body inside than human cells. How crazy is that? That there's more of these little things living inside of us than cells that make up our own body. So it makes you go, kind of blows the mind that, oh my God, there's this stuff all over the place. But don't freak out. It's good stuff. Most of them are helpful. We want to identify the bad ones, try to minimize them, identify the good ones, and benefit them. So you guys have seen commercials for probiotics. That's enhancing the bacteria that are living inside your intestinal tract. Pro means it's for, versus antibiotics, which mean against. Okay, what's a neat feature about bacteria? Well, we started with they're generally single-celled. Their size, that spectrum of size. Here's another feature, nucleoid region. Bacteria have DNA, just like us. There's actually some genes we share. But instead of their DNA being inside of that little ball we call the nucleus, it's strung out in a general region. They call it the nucleoid region. Let me reduce this a little bit. Okay, so here's a very, very poor drawing of a bacterial cell. I want you guys to draw one yourself. And the DNA is one big giant loop. There it is. This is the region where the DNA is found. That's what we call the nucleoid region. If we're looking at our cell, we'd have a nucleus. And that's where all of our DNA would be housed and contained. Okay? Now, the other really, really cool thing about bacteria, they have these little loops of DNA. Draw a couple little loops out here. So you're going to label this nucleoid region. There's my label there. I'm cheating. Write it out, label it. And then these little circles or loops are called plasmids. So label it. There's your they're plasmids. Plasmids are little loops of DNA. Now what's so cool is they can actually share that with another bacterial cell. So imagine sharing a piece of your DNA with somebody next to you. Hey, you want the gene for my blue eyes? Here you go. They can do this. I mean, it's crazy. This is why they're so adaptable. Maybe one of them has a gene for resistance to penicillin, and it passes that gene on. It puts the gene into a plasmid and hands it over to everybody else, and now all those bacteria develop resistance. How crazy is that for a very simple organism? So nucleoid region, very, very cool, neat feature that's special to the prokaryotes, bacteria and archaea. Reproduction and adaptation. All right. Reproduction is fast. So prokaryotes tend to be very small. Doesn't take long for them to reach full maturity size. They can go through a process known as binary fission. This is equivalent to what we would call the cell cycle. Trying to write, sorry. You guys know by now the writing is not very neat. It looks like a fourth grader on Red Bull. So binary fission is kind of their version of the cell cycle, but it's quick. That's the big feature, short generation time. So some bacteria can reproduce in 20 minutes. Every 20 minutes they're replicating and replicating and replicating and creating new bacteria. They can also do a thing known as horizontal gene transfer. So remember those little plasmids? Here's bacteria, we're going to call this A. And now we got bacteria B over here. And they're looking at each other saying, hey, what's up? How you doing? You like my gene? Here you go. And that one will insert its little plasmid through a little tube here, known as a conjugation tube, to the other bacterial cell. That's just phenomenal that you can pass genetics laterally within a population. For us to pass genetics, we reproduce and have another generation. You can't hand your gene to a relative, to a brother, a sister, a parent, or anybody living right now. You can't hand it to a living individual. You create a new generation when you combine the egg and the sperm. But these guys can hand genes back and forth across the population. Talk about adaptation. All right, so some really, really amazing things about these things. So again, I want you guys to start thinking about this. Do I like the bacteria? How do I enhance these traits, these features? Do I not like the bacteria? How do I stop them? How do I prohibit them from doing these things? Internally, their internal organization is different than a eukaryotic cell. So they don't have those little compartments, what we call organelles. They don't have the same little ball, the nucleus, the Golgi, the etc. Instead, their membranes are, their plasma membrane is kind of folded in, and it creates the respiratory membrane and a photosynthetic membrane. So this membrane here is literally the cell membrane of the bacteria that's just kind of folded inward, and this runs cellular respiration, producing ATP for the cell. This would be analogous to, you guys remember a little powerhouse of the cell? I'm not going to tell you, I want you to look it up. You're like, oh, dang him. Okay, over here, thylakoid membrane. Hopefully you can see the green. Same kind of idea. Their cell membrane is folded inward, creating a photosynthetic membrane. So this would be analogous to that structure in the plant cell that runs photosynthesis. Again, I'm not going to tell you what it is. I want you to look it up. Okay? If you get stuck, email me and I can help you out. But hopefully everybody, top of your head, you're like, oh, I know what that one is and what that one is. We're going to see those structures again very soon. So internal organization is a little bit different than the eukaryotic cells, right? One other thing to mention here is they often have little attachments called flagella, these little tails that will help provide mobility, swimming, moving, propelling them around. Some of them will have other little attachments known as pili, little hooks that enable them to attach on to other structures. All right, so you got the gist of the main features of bacterial and prokaryotic cells. In the second part of the lecture, we're going to talk about some differences between the bacteria and the archaea, what splits those guys apart and puts them into their own domains. All right, so tune into the next one.